Today we have the topic cartilage, which is the part of skeletal connective tissues. Do not forget that some students, due to holidays, have two topics together, cartilage and bone. So please check your timetable, and if you have your lesson on Monday or Tuesday, for sure you have those topics combined. So today we talk about cartilages. This is a very easy topic, so don't miss your chance to get the highest grade this week. We start this topic from the classification. There are only 3 types of cartilages in our organism, and for every subtype you have to know its typical location. First, let's distinguish the names: hyaline, elastic, and fibrous. There are 3 types. Hyaline cartilage could be found predominantly in the respiratory system, like in our nose, trachea, ribs, and of course in the articular surfaces of our bones, which is very important. They form joints. Then elastic cartilage could be found predominantly in our organ of hearing, in the ear auricle, in the auditory tube, in the external auditory canal, and in some other places. For fibrocartilage, the most well-known example is the intervertebral disc, and we'll investigate it in details at the end of the lecture. And now, as for the hyaline cartilage, how we can distinguish those 3 types? By the macroscopical analysis with an unarmed eye. Hyaline cartilage is bluish and semi-transparent due to a high amount of water. Up to 80% of its composition is represented by water. That's why it has its name. Hyalos means glass. Glass-like consistency, homogeneous and semi-transparent. Then, as for the elastic cartilage, you already know that elastic fibers, they have yellowish color when they are unstained, so elastic cartilages are also of yellow color. Easy to remember. As for the fibrocartilage, it has dense bundles of collagen fibers, and they could be seen even with an unarmed eye, so thick they are. That's why it has its name, fibrocartilage. And as scar tissue, it is of white color. Nothing difficult. Okay. Then, let's continue. Hyaline cartilage, in general, as for all 3 types of cartilages, we have to say that, of course, they have cells and ground substance, and cells for cartilage tissues, for all 3 types, they are divided into chondroblasts, which are immature and metabolically active, and chondrocytes, which are mature, differentiated cells, less active than comparing with the chondroblasts. What is also important to mention, that those cells, they derive from the mesenchymal stromal cells. You remember them from the topic connective tissue, and you remember that in connective tissue we have the population of stem cells, which are mesenchymal stromal cells, and they are classified as stromal or stem cells because they can give rise to fibroblasts, osteoblasts, chondroblasts, and adipocytes. So the possibility to give rise to a wide range of cells is the basis to call them and to classify them as a stem cell. So do not forget, mesenchymal stem cells are a very promising source for the regenerative medicine in future. So chondroblasts and chondrocytes, they derive from the same cell as fibroblasts or fibrocytes, but they are characteristic for cartilage tissue, and in some other places, under some circumstances, also inside the cartilage, we can find chondroclast cells. Their function is to resorb the cartilage, helping to replace it with a bone, but under normal circumstances we can find them. So today we don't talk about them. This is the topic of the next lecture. We'll return to them in the topic bone tissue. Okay, as for the matrix, around the cells, in all 3 types of cartilages, there are fibers and ground substance components. As for the fibers, please remember that type 2 collagen is characteristic for all 3 types of cartilages. So type 2 collagen is called something like characteristic for cartilages, because it is found almost only in the cartilage tissue. So do not forget that it is of 2nd type. not only the name collagen, but also the type of collagen. Actually, there are many more collagen subtypes like 9th, 11th, many others, but type 2 is the most abundant in the cartilage. Then also inside the matrix there are lots of proteoglycan aggregates, particularly aggrecan. Aggrecan, this is the type of proteoglycan with high amount of chondroitin sulfate and keratan sulfate. So it has a special ratio between the chondroitin and keratan sulfate, and it is called aggrecan. What is the proteoglycan aggregate? You should remember from the topic connective tissue proper. Please return to another lecture if you do not forget, if you do not remember. Then this matrix is distributed not uniformly, but instead as well as cells. And on the cross section, in the histological section, we can distinguish some regions, and let's investigate the specimen. So mature cells or chondrocytes, they are not distributed evenly, but they are grouped together, and such groups are called isogynous. Isogynous because they derive from the same maternal cell. Iso means the same, genus like generation, the same generation, because they are generated by the same ancestor, by the same maternal cell. And just after division, being divided, cells can't migrate far from each other because the matrix is very, very dense and stiff. So they stay together, and this is what we call isogynous group. Now, this group resides in the space called lacuna. Lacuna is a cavity, or this is like a cave in a rock or a cave in a stone in which cells are located. And this region, this territory, is filled with more viscous matrix, more soft matrix, comparing with the matrix around the lacuna. And inside the lacuna, just around the isogynous group cells, we have the territorial matrix, while between lacuna, between the isogynous groups, we have interterritorial matrix. They are called so because territory of the lacuna filled with a territorial matrix, and between lacuna, interterritorial. Inter means between, interterritorial matrix. And territorial matrix, which is inside the lacuna, it is represented by proteoglycans, highly sulfated and negatively charged, as well as some collagen fibers, of course. So this region is stained in more basophilic colors. So here we can see basophilic nature because of the negative charge of proteoglycans. While interterritorial matrix, it contains predominantly collagen type 2. So this space is more oxyphilic in its nature. So please do not forget that matrix is divided into territorial and interterritorial. First is soft and viscous to make a comfortable environment for chondrocytes. The second is dense and stiff to provide the cartilage with its stiff properties, because its main function is to provide mechanical skeleton. And also, except those two, there is one more, pericellular or capsular matrix. As you can see, just around the cells, the concentration of proteoglycan aggregates is the highest. So this is extremely negatively charged material, and this is what we call pericellular or capsular matrix. Okay, in general, we have considered the structure of the hyaline cartilage, and now let's move on. And also we have to add that on its surface, almost everywhere except the articular surfaces of our bones, there is a perichondrium. Perichondrium is a dense connective tissue with blood vessels and chondroblasts. And the most important function of the perichondrium is the regeneration and nourishment. Remember, due to blood vessels, it supplies the cartilage tissue with nutrients, and due to the presence of immature cells, they can stimulate it can regenerate the cartilage. Notice that cartilage itself, not only hyaline as well as elastic and fibrous, they are devoid of blood vessels, so the thickness is limited, as well as in epithelial tissues. And absence of blood vessels results in the special feature in the low regeneration. So cartilage tissue, unlike epithelial tissues, they almost do not regenerate. They regenerate very slowly and ineffectively, and usually they are replaced with a connective tissue scar. And it is important to take into account, so we have to preserve our joints and all our cartilages along the life, because it is very difficult to regenerate them. Okay, so perichondrium, this is the dense connective tissue on the surface of the cartilage, and it contains chondroblasts and blood vessels. In terms of perichondrium, we have to mention that cartilages, they have two types of growth, and usually they grow early after the birth and up to the puberty. Yeah, the active growth of the skeleton is accompanied first of all by the growth of our cartilages. So cartilages, they grow under the action of a set of hormones we'll consider later, and there are two types of growth. The first one is when cartilage tissue is added at the periphery, and this is what we call appositional growth, and it is provided exactly by the perichondrium. So here perichondrium it generates new cells, they enlarge, start to produce matrix, and being embedded in this matrix, they are transformed into chondrocytes now. Then, next type of growth is interstitial. Interstitial means from inside, when chondrocytes themselves divide very slowly, but they can divide inside lacuna and produce more matrix around them. And it is what we call the interstitial, when cartilage grow from inside. So appositional is provided by chondroblasts, interstitial by chondrocytes. Now, as for the hormone control, somatotropin, or the growth hormone, it stimulates the division of cartilage cells, predominantly in the perichondrium. So it stimulates the growth of the cartilage. Then also thyroxine and testosterone, male sex hormone, they stimulate the production of the extracellular matrix. That's why in boys, for example, along the puberty period, along with the sexual maturation, different cartilages, they enlarge and skeleton changes significantly. Larynx also enlarges, yeah, and it results in some voice changes. That's exactly due to the testosterone. Thyroxine is also important for the growth, so deficiency of the thyroid hormones might result in the problems with the growth as well. But you can see that the effect of those hormones differ from the somatotropic. Notice that cortisol and estradiol, they inhibit the growth of cartilage. So in girls with sexual maturation, instead, growth of the skeleton usually stops. And we return to this topic next lecture when we discuss the growth of bones, because growth of cartilage tissue is tightly interrelated with the growth of bones. Okay, so we have considered the structure of the cartilage, the process of its division, and now we have to mention that unlike growth, regeneration is very slow. Okay, I've already said that. Let's look at the specimen, and that's what you have to analyze and recognize under the microscope during the practical lesson. So in the hyaline cartilage, notice that cells are grouped in the isogenous groups. Here they are, the isogenous groups. Notice that inside the isogenous group, the matrix is much more basophilic, negatively charged, than the matrix between the isogenous groups. So here interterritorial matrix, which is more oxyphilic because collagen fibers are there, while inside isogenous groups the matrix is basophilic, and just around the cells it is especially basophilic because this is the capsular matrix. Notice that the interterritorial matrix made up of collagen, so it is stiff, and that's why cells, they can't migrate far from each other. That's why they stay and form isogenous groups. That's why they are not evenly distributed. Notice as well that at the periphery there is the perichondrium with a dense connective tissue and chondroblast cells. Here they are chondroblasts. You see they are flattened, they look like fibroblasts, and by division they give rise to chondrocytes. They start to grow, enlarge, produce more and more matrix. Eventually they become enclosed, embedded in this matrix, and they continue to divide, giving rise to the mature chondrocytes. Okay, that's what we can distinguish in the regular specimen. Now let's continue to investigate hyaline cartilages and its special subtype, articular cartilage. Articular cartilage is especially important because it is affected with aging, and due to mechanical overloads in some sportsmen, for example in football player, their knee joints are very often affected by different diseases due to mechanical overload. That's why we have to understand the structure of this cartilage and its features. Here it is written joints and diarthroses because there is also one more type, synarthroses. And from anatomy you know that in synarthroses two bones are connected and they almost don't move. But in diarthroses the bones are connected and they can move. This is the mobile joining of the bone, of two bones. And the surface of such bones, the surface of diarthroses, are covered with hyaline cartilage or articular cartilage. What is a special feature? The feature number one is that this surface has no perichondrium, neither on the surface nor on the border with the bone tissue, so it doesn't regenerate very well. The second feature is that the surface is covered not with epithelial lining nor with cartilage tissue itself, but instead there is a synovial membrane. Synovial membrane, it lines the joint cavity from inside. Usually we used to see epithelial tissue in such regions, but in joints we have synovial membrane, and here its structure it is given. Synovial membrane, it is made up of connective tissue and it includes two general types of cells. Type A cells, they are macrophage-like. They derive from monocytes and they provide phagocytosis of different debris formed in the articular surface inside the joint cavity. So macrophages, they regulate local inflammatory processes and they are responsible for the removal of rubbish out of there. Then fibroblast-like type B cells, this is the second type, and they produce extracellular matrix, particularly ground substance components, and like protoglycans, hyaluronic acid, and those elements they help to make the synovial fluid more viscous in nature, gel-like in nature, and it reduces the friction along with the movement of the bones. And also there are blood vessels. Blood vessels, these are the capillaries of fenestrated type. What about in details about fenestrated capillaries we talked during the last lecture in this semester, but now you have to know that fenestrated capillaries, they are perforated and blood plasma percolates easily through their wall, and in this way synovial fluid is produced. So synovial fluid is produced from the blood plasma via the filtration through the fenestrated capillaries, and some ground substance components are added by the fibroblast-like cells. So in the synovial cavity we can see this fluid, and under normal circumstances everything is very well because the surface is properly lubricated, the surface is smooth and clear, and the hyaline cartilage reduces the friction. But with aging, it is important to say that hyaline cartilage as well as fibrocartilage, they undergo calcification. Calcification is when calcium salts appear inside, and usually if the degree of calcification is very, very high, then it results with a replacement by bone tissue. So, for example, in our trachea with aging, instead of hyaline cartilage, hyaline semi-rings, bone rings appear, and this is the sign of aging. Okay, so as for the articular cartilage, we have to say that it not only has no perichondrium on both sides, it also has no blood vessels as other types of cartilages, and also it has a special orientation of fibers. There are 4 zones, and on the superficial zone, cells are flattened and oriented parallel to the surface, and this is what we call the superficial zone. It is also called tangential zone due to orientation of collagen fibers. Under it, we have the intermediate or transitional zone. Here we can see more rounded cells, a little bit larger in size. Here the transitional zone is. And after that, the thickest zone, which is the deep radial zone. In the deep radial zone of the articular cartilage, collagen fibers as well as cells, they are arranged along the parallel lines. That's why it is called radial zone, because here the orientation of fibers, they resemble rays. And the last zone is calcified zone. Calcified zone is located on the border with the cartilage tissue. And here the tide mark helping us to distinguish easily the border between the bone and the cartilage. And here chondrocytes, they undergo interstitial growth, they divide, and by division they provide the growth of the cartilage, and in this way slow regeneration process takes place, but very, very slow. Usually along with the regeneration, cartilage is replaced with a scar tissue. Okay, after we have discussed the hyaline cartilage, we need to mention some details and features of elastic cartilage tissue. First of all, of course, it has elastic fibers, it's obvious. But if we look at the specimen, we can find that isogenous groups here are arranged in 2 cells. They're very small, and usually there are only 2 cells, as in hyaline cartilage they are much larger. Then in the matrix between the cells, we can distinguish fibers if we apply special staining procedures like Orsin or Resorcinfuscin. Those fibers are thin and branched, and they are not dissolved in the water. That's why we can distinguish them. While collagen fibers, when they are very highly hydrated, they can be distinguished because they are thick and dissolved completely. Okay, on the surface of the elastic cartilage, we can see chondroblasts inside the perichondrium. Here we can see dense connective tissue fibers, and after that chondroblast cells. Chondroblasts, they divide, and in this way they provide oppositional growth and the growth of cartilage in thickness by adding the new tissue from outside. Here another view of elastic cartilage is given. Again, we have the perichondrium with the connective tissue fibers and chondroblasts and very prominent elastic fibers. As you can see in the matrix, also of course we can find collagen type 2. It is characteristic for all 3 types of cartilages, but elastin is more numerous here, and it is responsible for the general properties of such cartilages. Then, if we compare hyaline and elastic, we can easily distinguish elastic because, again, by the shape of the isogenous groups, you can see irregular shape or spherical shape here. And in elastic cartilage, usually isogenous groups made up of 2 cells only, and they are oriented almost parallel one with another. Then interterritorial matrix here is homogeneous, while there it is represented by very well distinguishable elastic fibers. Both they have the perichondrium here and there. Then elastic cartilage itself one more time. I think nothing to show. Again, the perichondrium, the cartilage tissue with chondrocytes and elastic fibers. Nothing new. And at last we go to the fibrocartilage. This is the last type, and as you can see, it has no perichondrium. Please remember the first feature. Unlike hyaline and elastic, it has no perichondrium. And in its matrix, except collagen type 2, we also can find collagen type 1. What is the feature of collagen type 1? From the topic connective tissue, you remember that this type, it forms thick bundles, and if other collagen molecules they form fibrils, then fibers, collagen type 1, it forms even bundles of fibers, so they are very thick, and that's why we can see them with an unarmed eye. For example, in the intervertebral discs, we can see the circular orientation of those fibers even without microscope. This is type 1 collagen. It is of white color and very thick. And of course, protoglycan aggregates. Notice that aggrecan, which is also present in the hyaline cartilage, we said that it is made up of keratan sulfate, chondroitin sulfate, and it is produced by chondrocytes. And also they have versican. Versican is the protoglycan produced by fibroblasts. It's very easy to remember that fibrocartilage, it has not only chondrocytes, but also fibroblasts. Fibrocartilage, it has fibroblasts. Fibroblasts produce versican, while chondrocytes, they produce aggrecan. Okay, notice that there are no chondroblast cells. There are no chondroblasts, there is no perichondrium. And if we look under the microscope, how we can distinguish this cartilage from others? It has a lot of parallel collagen fibers. Notice we can see them. They are so thick they can't be dissolved in the water. They are too thick for that. And the cells are arranged like a stack of coins along the same line, along the parallel lines. So isogenous groups, they might contain many cells, but they are elongated and they are oriented along the same axis. And here the intervertebral disc is given. Notice that here there is the bone tissue as well as there, so there are two vertebrae by each side, and in between the two vertebrae there is an intervertebral disc. Here it is. And in the intervertebral disc there are two components. There is an annulus fibrosus. You can see that collagen fibers are oriented in a circular manner at the perimeter, and in the center there is a jelly-like soft nucleus pulposus, central nucleus pulposus, which is made up of protoglycans, hyaluronan molecules, and it is used as a shock absorber. And sometimes due to its sometimes this results in the herniation when the nucleus pulposus shifts and when it goes out of the annulus fibrosus, this is what we call the herniation process, and it is very painful, and it results in the compression of the spinal nerves, and this is a rather severe pathology. So the structure of the intervertebral disc is very important, and this annulus fibrosus should be dense enough to hold the nucleus pulposus in the center, because if this nucleus pulposus moves out, then this is a very severe pathology. Okay, this is the last slide of our lecture, and as you see, you have to prepare a little material for the next lesson, so don't miss your chance. This is a very simple topic. Good luck.